This is a story about a little boy who grew up around the ocean. He loved reefs and corals, but was always told you're too young, too small, you'll just have to wait till you're older to go out to the reef on your own. Day after day, he came back to the beach, waiting for the day he'd be able to go out to the reef by himself. After 6 years, he was finally old enough to go to the reef alone. But by the time he got there was a sign posted saying, "Reef closed." He wondered to himself, do I have time to make a change, or has my time passed me by? He wondered, what could I have done? And he left searching for an answer. He searched beach after beach, until finally he found the scientist he was looking for.
Well, welcome to my lab. We're in Fort Pierce, Florida at Harbor Branch Oceanographic Institute at Florida Atlantic University. My name is John Reed. I'm a research professor at Harbor Branch, where I've worked for the past 40 years studying deep water reefs and shallow water reefs and mesophotic reefs and trying to protect and conserve them.
What are the major threats to corals?
Well, actually there are many different threats. Corals and coral reefs around the world are being threatened and impacted by a variety of sources, some man-made and some not. The primary threats are, number one, pollution. We have pollution from runoff from land and from rivers that go out into the ocean. The runoff from land can carry silt, and the silt can cover the corals. It can block the light. The siltation on the coral, it kind of clogs them up and also can clog up the gills of fish. The other types of pollution are nutrients, nitrates and phosphates. And those are especially bad on coral reefs because as they come into the ocean or estuary, these pollutants are fertilizers, so they fertilize algae and phytoplankton the same way as you put fertilizers on your lawn and garden to make it grow. Well, when you put excess fertilizers in the ocean, you have these blooms of algae. And what happens is the plankton is on the surface of the water, and it actually kind of filters out the sunlight, which is bad for the coral. You also have runoff of heavy metals and plastics and many things that aren't good for the environment. Coral disease is another major factor. Starting back in the early 1980s, we first started seeing these major disease outbreaks in the Caribbean and around the world, but in the Caribbean, it first started off with different corals getting different types of diseases where the tissue sloughs off. You have things such as black band disease, white plague disease, all kinds of diseases. We don't even know what they are, but typically they're from either bacteria or viruses. I was studying a deep water reef in the Gulf of Mexico in about 300 foot of water. And it's a deep water reef off of the Florida Keys, off Dry Tortugas. In 2010, we were there to study the oil spill, the big oil spill that happened. And we had a submarine, a little submersible, 4-person submersible diving along the coastline. And we went to this reef called Poley Ridge, and we saw 6 lionfish in 2010. Well, since then, I've been studying that reef every year from 2012 to 2015. And the last time we were there, we probably counted thousands of lionfish. I mean, thousands and thousands per square kilometer. So they're very prolific throughout the coastline of the southeastern United States and the Bahamas and throughout the Caribbean. And there's other species, invasive species such as algae and different things that once they get here, they don't have their natural predators, so they overgrow and take over the environment. And especially in Florida, we have many, many invasive species. So this is Lophelia coral. It's kind of similar to the Oculina in that it's a deep water coral. It has no algae growing in it. It's called azoxanthellate coral. And so it has to get all of its energy from capturing plankton from the water column. That's why it has such large polyps. It has large tentacles for grabbing plankton out of the water column. So, this is Oculina varicosa. It's a branching scleractinian coral, and this makes these amazing coral reefs off Florida. It occurs here and nowhere else on Earth. And off Florida, it makes these mounds that are 60 to 80 feet tall, kind of conical cones, and the mounds are covered with big bushes of coral up to 6 feet tall. Well, a little coral head like this one, like the size of a soccer ball, can have 2,000 animals living in the coral. It takes hundreds of years to grow. This coral here grows a ½ inch, 1/2 inch in a year. So to grow into a meter-sized coral head, you're looking at hundreds of years old. So they knock the coral down so they can come back with the nets and get the shrimp. So I worked with NOAA Fisheries and the Fishery Management Council for years. In 1984, was able to protect 90 square miles, and that was the first deep water coral marine protected area in the world.
Next, I went to see David Burdick, a research associate with the University of Guam Marine Lab and coordinator of Guam's long-term coral reef monitoring program.
I was born in Conneaut, Ohio. It's a small town in northeastern Ohio, right on Lake Erie. So I grew up near the water, and I think that's where my fascination with the aquatic environment and aquatic life was formed. And from there, I had some experience in the Bahamas, and that was after I got my bachelor's degree in biology, when I knew I wanted to study life, to be a biologist. I realized through my experience in the Bahamas that coral reefs are not what they should be. And unfortunately, the human communities that are reliant on reefs are affected by the decline in reef condition. So that led me to reconsider my plans to becoming a kind of part-time biologist, part-time beach bum, and actually pursuing a graduate degree. My primary responsibilities are to coordinate Guam's long-term coral reef monitoring program, which is funded by NOAA. But then the other half of my job, and this is a relatively recent thing that happened after I moved to the University of Guam, is to manage what's called a biorepository. And this is a National Science Foundation project, or part of a project funded by NSF, where we're aiming to bring together and expand upon the existing collections, biological collections rather, that we have here at the University of Guam, but then also to digitize them and make records, images, and perhaps even 3D images, 3D models available to researchers across the world. The last part of that question is what keeps me passionate about photography. That's another one of those things where it kind of started out small, where I just took a few photos here and there in the Bahamas, even with disposable cameras, and took some pretty good shots. Thought I was pretty good at it. It was fun. So why not stick with it, right? And then from there, you know, there's always a technical challenge, depending on, you know, what kind of camera you have, the lighting environment, other abiotic conditions that you're shooting in, and then the organisms themselves require different skills. So taking a good picture of, say, a sea slug is much different than taking a picture of a fish swimming by you fast. The Marine Lab here, I've been able to work with a group called SECOR, which is an acronym for Sexual Coral Reproduction. So I think SECOR International is technically their name. And so they're a group that they're dedicated to doing research, then ultimately employing coral reef restoration techniques that revolve around sexual reproduction and the propagation of corals that are produced from sexual reproduction. So instead of breaking off a piece of coral and outplanting it somewhere else, so taking fragments or fragging the corals, they're actually collecting the egg sperm bundles, or depending on the species, the eggs and the sperm, and then mixing them together from different colonies and producing coral larvae, excuse me, growing them out to a size that kind of gets past the survivorship bottleneck, where a lot of small corals, newly settled corals, die.
Then I went to see Peter Etnoyer in my own backyard, Charleston, South Carolina.
My name is Dr. Peter Etnoyer. I am a marine biologist for NOAA. I was born in Philadelphia, Pennsylvania. My favorite kind of coral is a deep sea bamboo coral. I like that coral because it glows in the dark and because I discovered a new species of it back in 2004 on a seamount in the Gulf of Alaska.
As a kid, I know you dove. What were the corals that you saw then like compared to the ones you are seeing now?
I started diving around the age of 14. I did my first dives in New Key, Florida, and the kind of corals I was seeing were large, massive corals, brain corals, and also sea fans and branching corals. And the reefs were very healthy and colorful back then. There were quite a few fish. These days, the reefs around Florida are a little less colorful and there's fewer fish. And we expect that's a result of coastal development, eutrophication of the waters, and also climate change.
Who was your greatest influencer?
My biggest influencer in my research was Jacques Cousteau. He had a television series when I was a young kid. It was in black and white, but he would go out, strap on a scuba tank, and explore coral reefs around the world and talk in a funny accent about things that he was saying. It captured my imagination, but also educated me about this unseen world that was just offshore.
When you're looking through a tiny screen on a robot in 1,500 feet of water, looking for deep sea corals in the Gulf of Mexico, how do you determine what appears to be significant to your study?
When I'm in the field using a robot to survey the deep sea, and we're conducting video surveys or photographic surveys, what I'm looking for is the size of colonies, the health of colonies, and the abundance of colonies. Those three things pretty much tell me the health of the broader ecosystem. Size is correlated with the age of the colony. The color is associated with its health. If it's brightly colored, it'll be a healthy colony. If you see brown tissue or dead branches, then it's not doing so well. And the abundance of the colonies tells you how favorable that particular habitat is to that species of coral.
What is the most significant exploration you have been a part of?
The most significant ocean exploration I've been a part of was one of my first expeditions aboard a ship with the Alvin submersible. This was back in 2002 and 2004. We were doing the first ever explorations of a dozen seamounts in the Gulf of Alaska. These seamounts rose from a base of around 4,000 meters deep up to around a peak of about 500 meters deep. So it's a submerged mountain covered with life, corals, sponges as large as I am, and abundant fishes and squids and octopus.
If you were designing an exploration of corals, what would it look like?
If I was designing a new exploration of corals, I would target several areas at similar depths with similar seafloor character, but separated by various distances. And then I would do multiple surveys, ROV dives, and transects over those features in order to compare them to see if they're the same or different.
What do you think would help further address corals dying?
In order to further address the demise of coral reefs around the world, we need to get a better handle on the amount of fishing that's taking place in those areas, the type of fishing that's taking place, whether it's destructive or non-destructive, and we have to also understand the amount of offshore runoff or pollution that's coming from land to the nearby areas. These are very sensitive animals. Any kind of toxins or detergents in the water would likely make that water inhospitable.
What advice would you give to future generations?
If I was going to tell one thing to future generations about how to conserve and protect our ocean environment, it would be to get out there as much as possible, to make sure that you experience this environment so that you can appreciate it and that you can share it with others. If I was going to say a second thing, is to remember that conservation starts at home, and you have to live the way that is sustainable for you and also share those experiences. So get out there, enjoy the nature that you love so much, and also remember that everything you do at home can have an impact on those environments. So you have to, walk the walk and talk the talk.
Thank you.
Sure not.
Next, I went to speak with South Carolina Congressman Mark Sanford in Charleston, South Carolina.
How, if at all, or has the government supported coral rehabilitation in your eyes, positively or negatively?
Well, on the positive side, I voted for the Coral Reef Protection Act, or what it was. I think that was the name of it, back in 2000. Clinton signed it, and it afforded additional protections, at least within Virgin Islands, Hawaii, and Puerto Rico, in terms of protecting our own coral reefs. I think that's what we've done on the affirmative side. I think that the NOAA and the National Ocean Service and some of the related government entities have looked for ways of doing more. I think, particularly on the NOAA side, there is important work that's being done on a consistent basis to look at how coral reefs are doing based on warming trends in terms of temperatures nationwide, because they're apparently very, very fragile. If you look at what's happening in the Great Barrier Reef right now, things are coming undone that people never thought would be so. And this bleaching out of reefs is sort of nature's last cry. I think that on the research side, our government's done a fair bit. I think there's been specific legislation that has been helpful, though certainly not comprehensive from a global standpoint. I think what we've done to hurt, in as much as we're trending back toward more in the way of fossil fuel dependence, I would suggest that it doesn't help. The idea of opening offshore waters initially for Florida, that's no longer the case, I think would have been problematic in terms of some of the reefs down that way. I would say, the pendulum swings. It depends on the administrations. I wouldn't say that this one is particularly sensitive on the ecological front. That can mean what is going to be done next in ANWR, a giant refuge up in Alaska, will have real implications for the tropics. And so we live in a diverse, from a biological standpoint, world, but it's an amazingly interconnected world, and there's surprisingly little variables that can have a big impact from thousands of miles away. And so I would say some of what we're doing, opening up things like ANWR, I would see as working against the long-term viability and sustainability of our reef system around the world.
What is the process for approving a new law or environmental protection?
Well, it depends on what side. So if you're talking about environmental protection from a regulatory standpoint, an agency will go out and propose a regulation. Then they have a public comment period. We're going through this with the offshore question, offshore South Carolina right now. So there's, a proposal. Then there's a public input period. I'm going to be at a rally tomorrow in Columbia before I go back to D.C. And right before the Bureau meets and has their public visit for South Carolina, right before that, there's going to be, a rally of some sort in the State House steps. And I'm going to speak at that. So public comment, and then finally is the rulemaking. That's sort of the step on the regulatory side. On the legislative side, it's the squeaky wheel process I talked about earlier. So enough people make enough noise that a bill gets introduced in the House or the Senate, depending on who you're talking to, and then it's subcommittee, it's full committee, it's full floor vote. Then it's got to occur on the opposite side, the House or the Senate, as the case may be. If it's revenue generating, it has to originate in the House. But as it relates to coral reefs, it could be really either side. And you end up with a companion bill in the House or the Senate. Make its way through the process, then it goes to the President's desk, and then it's either vetoed or not. It's a much longer process on the legislative side. That's why a lot of environmental groups prefer regulatory change. It's one-stop shopping, it's quicker, and it can be done by legislative edict.
As we move forward, we need to remember that it is our time to act. It is important to remember our responsibility to not only the ocean, but to ourselves to protect these coral reefs that are dying. Corals provide so much for society that it is our duty to give back by being the voice that they don't have and advocating for them. A special thanks to Ruth Gates, Bill Arnold, John Reed, David Burdick, Peter Ednoyer, and Congressman Mark Sanford for their help and participation in making this film possible. If you would like to make a difference, please follow the link below and donate to the Kohala Center in Hawaii. They work diligently to help restore natural forests and protect coral reefs. Thank you, and remember, today is the day to act.